Hello my juniors, welcome to another biochemical lecture series. It's your great scientist Lorenzo, and today we'll be treating and dissecting the pyrimidine metabolism. So if you have not watched our video on chemistry of nucleotide, make sure you watch it before you watch the pyrimidine metabolism. If you are just new to my video and you're watching it for the first time, make sure to subscribe and turn on the post notification button so that when I drop any video, you'll be sure to be notified. And if you want me to make a video for you, just make sure to type it down in the comment section. When I go to it, I'll make the video for you. So let us jump right into the video. So today we're talking about pyrimidine metabolism. So pyrimidine metabolism simply is a metabolic or cellular process in which we form what pyrimidine bases, pyrimidine molecules needed for nucleic acid synthesis. What are nucleic acid synthesis? Formation of DNA and what? RNA. So simple. So because nucleic acid synthesis, which is what DNA and RNA, you need what? Polynucleotides. You need polynucleotides from purines and pyrimidines. So purines and pyrimidines are nucleotides. So in this video we'll be talking about only pyrimidine, not purines. Now what are the pyrimidine bases now? The pyrimidine bases include what? You have uracil, you have cytosine, and you have thymine. Uracil, what? Cytosine, and we have thymine. Don't forget. So you remember CUT, 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 C for cytosine, U for uracil, T for thymidine. So now cytosine. Now what is the structure of cytosine? Very simple. Cytosine molecule is what? 2-deoxy 4-amino what? Pyrimidine. 2-deoxy 4-amino what? Pyrimidine. Very easy. And the product here in this pyrimidine metabolism form is CMP. Don't forget CMP. The next one is what? Uracil. Cytosine is present in both DNA structure and RNA structure. Both DNA structure and RNA structure. The next one is what? Thymine. In thymine molecules, thymine is only found in DNA. Only found in DNA. So in this DNA molecule, what is the structure here? The structure is what? 5-methyl what? Uracil. 5-methyl what? Uracil. Or you can also say it's 2,4-deoxy. 2,4-deoxy 5-methyl what? Pyrimidine. That is the structure of thymine. The last but not the least is uracil. Uracil can only be found in RNA structures, and the chemical structure here is what? 2,4-dioxy. 2,4-dioxy what? Pyrimidine. Very easy. So once you understand all these molecules, we want to form these molecules in this reaction. So we'll be forming CMP, UMP, and TMP as the product of pyrimidine what? Metabolism. So don't forget it. Now the advantage of pyrimidine metabolism here is that in pyrimidine metabolism you form the sugar, you form the structure first, you form the pyrimidine heterocyclic ring first. You first form the pyrimidine heterocyclic ring first before you now attach the ribose phosphate sugar attached to it. Unlike purine, in purine you will form the ring in the sugar. You form the ring in the sugar. But in pyrimidine you form the ring first, then you now combine the ribose phosphate sugar attached to it. So that is the difference here. So since we understand that, now it is a heterocyclic ring, just one single ring, unlike purine that has double ring. So since we have said this one, now let us dive into the metabolic synthesis of it. How does it start and how does it end? So in the biosynthesis of pyrimidine, so stay with me, the biosynthesis of pyrimidine begins with the formation of what? Carbamoyl phosphate. The formation of what? Carbamoyl phosphate, or the synthesis of carbamoyl phosphate. So how do we form carbamoyl phosphate? Very easy. Now you see that carbamoyl phosphate can also be formed in urea cycle too, but this one is different because the carbamoyl phosphate formed in urea cycle is present in the mitochondria, while this pyrimidine ring formation, which is pyrimidine biosynthesis, occurs in the cytosol. So that is the difference. So now in carbamoyl phosphate we are condensing glutamine. We are condensing glutamine. So glutamine is donating the ammonia group that we are using here, and we are also combining with bicarbonate, which is donating CO2, and we are utilizing ATP. So when we condense glutamine, donating the amino group, bicarbonate donating the CO2, and utilizing ATP, also using magnesium as the coenzyme and also hydrolyzing the reaction, we'll be getting what? Carbamoyl phosphate. We're getting carbamoyl phosphate. So glutamine plus bicarbonate, utilizing ATP, using magnesium as the coenzyme, and also hydrolyzing the reaction, will form what? Carbamoyl phosphate. So carbamoyl phosphate is catalyzed by the enzyme what? Carbamoyl phosphate synthetase 2. Carbamoyl phosphate synthetase 2. Don't forget. Like I told you, it occurs in the cytosol, but carbamoyl phosphate synthetase 1 occurs in the what? In the mitochondria. So this is different. And also, since it occurs in cytosol, you don't need N-acetyl glutamate here. Don't you need N-acetyl glutamate as the allosteric what enzyme here. But this enzyme is a rate limiting enzyme, so it's only inhibited by UTP, uridine what triphosphate. Don't forget it. So now we have formed what? Carbamoyl phosphate now in this reaction. So that is the first reaction that you need to what proceed the reaction to the next level. So after the formation of carbamoyl phosphate, now you will condense the reaction. Now you condense the reaction with aspartate. Now if you condense the reaction or you transfer aspartate to carbamoyl phosphate, what are you going to form? You form what? Carbamoyl aspartate. This is a rate limiting enzyme, don't forget. The first reaction, carbamoyl phosphate, is inhibited by UTP. It's inhibited by UTP. So this one now, it is also a rate limiting enzyme, because when you condense aspartate with what? Carbamoyl phosphate, you get carbamoyl what? Aspartate. And what is the enzyme catalyzing it? Aspartate or transcarbamoylase. Aspartate or transcarbamoylase. Are you getting me? So what we call it is an oligomeric what enzyme. Oligomeric what enzyme, and it's inhibited by CTP and UTP. Cytidine triphosphate and uratidine what? Triphosphate. Don't forget it. So this is the step 2. The step 2, we are forming what? We are forming carbamoyl what? Aspartate. When aspartate condenses with what? Carbamoyl phosphate, using what? Aspartate or transcarbamoylase. Don't forget. So we have formed carbamoyl phosphate. The next one will form carbamoyl aspartate. So what's the next reaction? In this reaction, so simple. The same thing now. The ring now is closed hydrolytically. You remove water from the ring, you dehydrate the ring. So when you dehydrate the ring, you close it. So that's now we have formed carbamoyl aspartate. So carbamoyl aspartate is a ring that is open. It's a pyrimidine ring that is open, so we are closing it by dehydration. So when we dehydrate carbamoyl aspartate, we form dihydroorotate. We form what? Dihydroorotate, or some textbook we say dihydroorotic acid. Dihydroorotate. So what enzymes are we using? We are using what? Dihydroorotate. Dihydroorotate, removing fumarate from the reaction. So remove fumarate. So fumarate will be donated into the TCA cycle for what? Energy production. So don't forget. So carbamoyl aspartate is dehydrated with dihydroorotate to form what? Dihydroorotic acid or dihydroorotate. So now the ring is closed hydrolytically. So after now we have formed dihydroorotate or dihydroorotic acid, the next thing now will oxidize the reaction. So oxidize by utilizing what? FAD plus. We utilize by using FAD plus. So when we utilize FAD plus, we convert or we catalyze by what? Dihydroorotate dehydrogenase. In this dehydrogenase enzyme, remember, we are oxidizing the action, so we will reduce FAD plus to what? FADH2. Don't forget, if we oxidize the reaction from dihydroorotate, we will form orotate or orotic acid. Don't forget. The next thing is what? Transferring PRPP, which is phosphoribosylpyrophosphate pyrophosphate, to what? Orotate. So if we transfer PRPP to what? Orotate, we will getting what? OMP, which is orotidine what? Monophosphate. Orotidine monophosphate. Utilizing what? Orotate what? Phosphoribosyl transferase. Orotate phosphoribosyl transferase. So where do we get PRPP? Phosphoribosyl pyrophosphate. It's gotten from ribose sugar. So I tell you, HMP shunt, hexose monophosphate shunt, is metabolized what in an oxidative and non-oxidative phase. In the oxidative phase, what are you forming? You are forming what? Ribose 5-phosphate. Ribose 5-phosphate undergo isomerism to form ribose 5. So ribose 5-phosphate now utilizes what we call phosphoribosyl kinase. Phosphoribosyl kinase to form what? PRPP. Don't forget it. So that PRPP will form now is transferred, is utilized in purine and pyrimidine metabolism. Now after OMP, what do we get? Now OMP will be CO2 will be removed. It will be decarboxylated to form what? UMP, which is uridine monophosphate. OMP, uridine monophosphate, is decarboxylated to uridine monophosphate by what? OMP what? Decarboxylate. OMP decarboxylate or uridine monophosphate what? Decarboxylase. Don't forget. So first of all, from orotate we accept PRPP to form what? OMP. OMP to UMP we use the decarboxylase enzyme. So now one thing you have to know that the combination between orotate phosphoribosylate transferase and OMP decarboxylate, they are grouped. They are multi enzymes. They are called what? UMP synthase. So both of them are called UMP what? Synthase. Now we are from UMP. Now UMP is the first RNA molecules, the first product of what? Pyrimidine what? Metabolism, the first product of pyrimidine metabolism. Then from UMP we can divert to do anything we want to do. From UMP, first of all, from UMP we utilize what? Nucleoside monophosphate kinase. So when we utilize from UMP, if we utilize nucleoside monophosphate kinase, or we go straight to nucleoside diphosphate kinase, what are we doing? We want to phosphorylate UMP, the uridine monophosphate, twice. If we utilize it, first of all, we utilize ATP. The first ATP we are using here is what? Nucleoside monophosphate kinase. We'll get what? UDP. Don't forget. So when we utilize 1 molecule of ATP first, what are we doing? Converting ATP to ADP. We'll convert what? UMP to what? UDP, using the enzyme nucleoside monophosphate kinase. So when we do it again, we use nucleoside monophosphate kinase again. Nucleoside monophosphate kinase again. What are we doing? Then we will now add another, we will now utilize another ATP to form ADP by forming UTP. So if we went from UMP to UDP to UTP by using what? Nucleoside what? Monophosphate kinase twice. Don't forget it. So now we have utilized 2 UTP at this reaction. So now we are from UTP. From UTP now, what are we going to use? We will now use CTP synthesis. We will utilize CTP synthesis. So from UTP we can form CTP. For UTP we can form CTP using what? What is CTP? Cytidine what? Triphosphate. We can use uridine triphosphate to form cytidine triphosphate, utilizing what? Cytidine triphosphate what? Synthetase, utilizing what? ATP. Don't forget. Again, UTP to UCTP, using what? CTP synthesis, breaking down what? ATP. So here we are utilizing energy. Now we have got the CTP. From UMP2 we can also go to formation of TTP. So first of all, we have UMP. First of all, we have to convert UMP, which is an RNA, to dUMP, which is a DNA molecule. So don't forget. Before we go to form TTP, remember we said thymidine is a DNA molecule. So, and we have formed CTP, which is an RNA molecule, UTP, which is an RNA molecule. So from UMP we want to form dTTP, which is a DNA molecule. So thymine doesn't occur in RNA, but it occurs in DNA. So how can we go here? First of all, we have to convert UMP to dUMP, which is what? Convert RNA part of UMP to DNA part of UMP. So how can we use? We utilize what we call ribonucleotide reductase. So when we utilize ribonucleotide reductase, we are converting an RNA molecule to a DNA molecule. So now we are converting UMP to dUMP. The ribonucleotide reductase we use in this reaction, we use to convert UMP to dUMP. What is it reducing? The reductase enzyme, and it converts thyrodoxine, the reduced form of thyrodoxine, to the oxidized form of thyrodoxine. So when we use this reductase enzyme, thyrodoxine is being oxidized. Then now from the oxidized form of thyrodoxine, NADPH is now reduced. NADP plus is reduced to NADPH in the same reaction. Remember, we oxidize thyrodoxine, so thyrodoxine is now reduced back for utilizing what? NADP plus to NADPH. Don't forget. Now we have formed what? dUMP. What will we use now? From dUMP we can now form dTMP. dTMP, this reaction is very, very important, especially in various in utilization of folate here, folic acid. Also to destroy the DNA, to prevent replication of bacteria, viruses, we also must inhibit this process if we want to do. Even cancerous cells too, replication can be prevented because if you form dTMP, you can form a DNA molecule. From a DNA, replication can continue occurring. So in this reaction is very, very important. So from dUMP to dTMP, which is uridine monophosphate to dithymidine monophosphate, what happens? You are using what? The thymidine synthetase in this reaction. So thymidine synthetase is catalyzes the conversion of what? dUMP to dTMP. Now in this same reaction, what are we using here? We are converting what and trihydrofolate N5N10 methylene what trihydrofolate to what dihydrofolate in this reaction. Don't forget to. Now we are converting N5N10 methylene what trihydrofolate to what dihydrofolate in this reaction. You can utilizing the pyrimidine what synthetase. Don't forget. Now the dihydrofolate here in this same reaction, the dihydrofolate is converted to what trihydrofolate using what dihydrofolate what reductase. Dihydrofolate what reductase in this same reaction, like DHF. So dihydrofolate gotten from N5N10 trihydrofolate is converted to dihydrofolate catalyzed by pyrimidine synthetase. And now dihydrofolate is now uses the catalysis of what dihydrofolate what reductase to what trihydrofolate. Now serine is converted to glycine, dropping off the methyl group to convert trihydrofolate to what N5N10 methylene what trihydrofolate, utilizing the conversion of serine to what glycine. So if you want to inhibit most drugs and the antifolate drugs here, if you want to inhibit the progression of DNA, you just have to either inhibit pyrimidine synthetase or dihydrofolate reductase, so that the reaction, because if the reaction keep continuing, what happens? You keep forming DNA, and when you keep forming DNA, replication will occur. So that's why some cancerous some carcinogenic agents or chemotherapeutic agents are also used in this reaction to prevent continuous formation of DNA molecules that can cause continuous replication. So now we are forming dTMP now. So now we have formed dTMP. What can we do? We can also use nucleoside monophosphate kinase. So when we utilize nucleoside monophosphate kinase, what happens? We'll add one phosphate, we utilize ATP, converting dTMP to dTTP. So when we utilize another kinase reaction again, we convert dTTP to dTTP. You get it now? So we have converted from dUMP to dTMP, then from dTMP to dTDP. The last one, we now use another kinase to form what? From dTDP to dTTP. So don't forget it. You can also convert our CTP to dCTP. So you just know that anytime you want to convert an RNA structure, which is CTP, UTP, and TTP, if you want to convert them to DNA, again UTP, CTP. If you want to convert them to DNA, what do you use? Ribonucleotide or reductase. Don't forget it. So some agents can be used. I told you in the pre that some agents, they are chemotherapeutic agents which you use to inhibit pyrimidine synthetase. And when you inhibit pyrimidine synthetase, you cannot convert dUMP to dTMP, and you cannot form DNA. You cannot form DNA structure. So pyrimidine will not be stimulated. So adenosine, deoxyadenosine monophosphate or triphosphate cannot even bind or cannot couple with T, because remember adenine binds with thiamine. So if there is no pyrimidine, which means adenosine DATP, the D-adenosine triphosphate cannot bind to D-thymidine triphosphate. So what will happen? You will prevent replication. So now sometimes to prevent proliferating cells or bacterial cells, what do you happen? You have to inhibit pyrimidine synthetase. Like I said, either you have chemotherapeutic drugs, so either we are inhibiting it or we are inhibiting dihydrofolate reductase. So sometimes we can use some agents, some chemotherapeutic agents like 5-fluorouracil and 5-fluorodeoxyuridine. So these two agents, once they enter, when you administer them, they are converted to 5-fluorodeoxy or uridylate. So when they are converted, they inhibit thiamine synthetase, which means the cell there will be no proliferation, and it will only be dUMP that is formed. When dUMP is formed, the cell do not recognize dUMP. So dUMP is just usually used. Uridine is usually used as RNA, not DNA. So now the next one is also, you can also inhibit dihydrofolate reductase, which is used in conversion of DHF, which is dihydrofolate to dihydrofolate. So you can use drugs like methotrexate. So methotrexate, aminoprid, and trimethoprid. They are folic acid analog used to prevent the formation of THF. THF is dihydrofolate. So when you prevent THF, what happens? no replication can also what occur, because when you form THF uses serine to glycine to form what N5N10 methylene what THF, and which it keeps the formation of dTMP going. So don't forget this. So I hope it all made sense and you have you understood all the steps I gave in the formation of our pyrimidine synthesis. How we formed OMP, UMP, CTP. Then from UMP we use ribonucleotide reductase to get dUMP, and finally dTTP. I hope it was easy, and just watch it over and over again. It will enter. All this thing needs repetition. When you repeat it over and over again, then to stay in your head. If you are new to my channel, make sure to subscribe and turn on the notification button so that anytime I drop any video, you'll be sure to be notified. Well, I will be moving into the regulation and the clinical and the disorder state throughout the course of the video. Bye bye.